Evolution may be in both directions, right? So mutation may make any species modernize, more advanced, or it can make the retrogressive as well. So like mutation are of both kind, like harmful as well as beneficial. So gene silencing means if there is any genes of any disease, we can make it silent, right? That is beneficial for us. Identifying the mutations which cause the specific disease, analyzing the pattern of genes disease. With the population, scientists have developed one very useful laboratory tool which allow them to detect the thousands of active genes at the same time. Microarray, right? Now let's understand what is the microarray. Using microarray, when the genes are active, they express messenger RNA, mRNA. We know that transcription takes place in active gene and mRNA formation takes place is produced from active gene and use a template for production of amino acid chain, means translation, on the surface of ribosomes. If there is a mutation in gene, a different form of mRNA will be produced. However, a gene contains very large number of the DNA bases, and there are many places where the mutation can occur. A microarray is tool which scientists use widely to show if a DNA sample from the individual contain any mutation. These samples are known DNA chips. Got it?
Yes.
So to the DNA chips, that they are just DNA samples which taken from a person or any organism. Basically, a DNA microarray is a slide on which there are thousands of spots, each spot in a specific position and contains a known DNA sequence. The mRNA samples are then collected. Scientists usually use a reference sample with known gene sequence and experimental sample. Typically, this will be from an individual with a particular disease which may have a genetic element or example for a form of cancer, right? That is also a type of mutation. The reverse transcriptase enzyme convert the mRNA into cDNA, means copy DNA, right? So cDNA, which is the DNA copy of the DNA which is made on the mRNA, right? Each sample given fluorescence label. Usually known sample is given clean fluorescent label and the experiment sample is given a red fluorescence label. The labeled DNA samples are mixed together and applied to microarray slide where they bind the matching DNA probes. This process is called hybridization, right? After hybridization, the microarray is scanned to measure the fluorescence light produced in different spots. If both samples are expressing the gene equally, the light will appear yellow, a mixture of red and green light. If the experiment sample expressing more than the control, the spot will appear red. If the sample expressing less than the control, the spot will appear green. We can collect a very large amount of the data using this technology. The analysis of the data provide detailed information about the gene profile. This causes for many diseases and the effectiveness of some treatment. That is called DNA array or gene array. What is microarray?
Yes.
Microarrays and breast cancer. Some form of the breast cancer respond very well to treat the drugs which block the estrogen receptor. This is this breast cancer is increasing frequently in the women, and basically in those countries where the country are more commercial or that they are more capitalized country, because the females do not prefer for the breastfeeding after the birth of a child, or rather than right, they don't give a birth of the child. On that time, the formation of some lumps takes place inside the mammary glands, and that causes the breast cancer. So this is one of the most frequently spreading disease right now in modern days. So some form of the breast cancer responds a very well treatment with the drugs which block the estrogen receptor or prevent the estrogen synthesis. Estrogen, you know what is estrogen?
Yes,
What is that?
it's a hormone, female hormone.
Hmm. Right, female hormone. Estrogen and progesterone, they are the two basically female hormone which are responsible for the. Okay, you remember that very good. Doctors can cause microarray to show the level of expression of gene for the estrogen receptor in individual patient. If the high, then patient will be respond well to these drugs and they will increase the chances of successful treatment. If the patient shows low level of expression of gene, the estrogen receptor, then the estrogen blocking drugs will not be effective and are given and are not given. This causes a different approach to treat the treatment must be used. The same thing is represented over here. mRNA, cDNA, right? Again, these both of the cDNA, green color, red color, both are the mix. Then the color may be yellow color. One both type of the cells not present. Normal cells and disease cells, right? Red color spotted in the disease cell. Now come to the bioinformatics. This is also a recent branch of the biology, which have just discovered. I think it should be in the trend since last 10 years only, most probably. I listened this term in the 10-year back only in the corporate world that there is some job for the bioinformatics. Now, what is the bioinformatics? DNA technologies such as macroarray, DNA sequences, DNA profiling generate a huge amount of data for more than was ever produced in the past during the scientific investigation. Individual scientists or even teams of the scientists could not possibly analyze these data to search for the pattern. This is why a new science of bioinformatics is so important. Bioinformatics is the development of software and computing tools need to organize, analyze raw biological data. This includes that the development of algorithm, mathematics model, and statistical test, which can help us to interpret the enormous quantity of data that is generated. Using the bioinformatics, we can process and use the uh, information generated using microarray and other forms of DNA analysis. Bioinformatics enable the scientists and doctors to use the information to form these DNA technique to adopt the care individual patient and learn more about the biology of many different organisms. So that is the application of the biological data and formation of software or tools. Now, benefit and risk of GMO. So before going into this PDF, I want to tell you what is the risk. So can you tell me, just first you tell me, is there any benefit or risk of GMO? Can you tell me?
Genetically modified organisms.
Yes. What is the benefit and what is the risk? I want to know from your end. I think
They can
nowadays,
help.
I don't know like what is the status at your country, but if we take average across the like Asian countries or even including the Europe and America, more than 50% the crops which we are right now we are consuming, I think they should be GM. And I think the 90% of the crops right now we are using, 90% of the food we are using, that is basically either hybrid or GM. Hardly we are taking any wild varieties. That is the condition right now.
90% of what we're eating is a GMO.
Either hybrid or GMO.
So is that risky?
This is really risky, and that is a matter of conflict between the two class of the biologists. There are one class of the biologists which want that the world population increasing day by day, and we need more food. The land is limited, and we know we need more and more food. So yes, the land is limited. Isn't correct?
Yeah.
And every day it is reducing the agricultural area. Reducing
Yeah.
every day. Cities are expanding. The agricultural land is reducing. Now, what else you can do? You can't increase the land area. So what you will do? You can increase the productivity of the same land. Right? 2 generations back, we used to grow 100 ton grain from that land. Nowadays, we are, with the help of fertilizer technology and genetic modified crop, we are growing 500 ton from that same land. Right? So we are increasing the productivity with the same land, and that's what you can do. Another group of the scientists, what they are saying by this way, one day you will lose all the original biological stock which were present in our country and in our land.
Yeah.
And previously there are certain certain varieties of the wheat that I know. There are certain variety of the wheat where just what we are doing, where uh, you need not to any don't need to give any insecticide pesticide. They have self resistance variety. You don't require any kind of the application of the biotechnology or any kind of the application hybridization. They are self resistant varieties, but we are losing those varieties because they are getting polluted because of GMO, because of hybrid crops. So that is a threat as well, right? And nutritional value, taste and all that you're losing. That I can feel.
So it can cause diseases.
Yeah, if it will, you have a certain recipe, and nowadays, after the, your grandmother have a certain recipe. Think it this way:
Okay.
your grandmother have a certain recipe to make a certain kind of the bread. Then your mom came, then you came, then me came, and all that. What we did, we are altering that recipe. We are mixing something. We have mixed up some Western food taste, then some Chinese food taste, some Indian food taste, and all that, right? Now the generation will go on, our child will come, we will become grandfather and grandmother, and our grandmother and mother, they will be no more, right? After some time, after 100 years, right? Or after 50 years, right? That generation will be wiped out. Now what grandmother used to do, what grandmother used to make, you'll forget all that recipe. Is that present? No. Because what we have altered that much that we forget what she told us. Fine. We forget what was the logic behind it. We forget everything, right? We forget that the grandmother used to make more nutritional. He was more nutrition oriented. That was more authentic. That was more nutritional. Now we are taste oriented. So wild variety will be wiped out in the same way. The same thing will happen with the original crop, right? Original varieties, right? We will require more and more insecticide, pesticide that will harm our health. Got it?
It would harm.
Definitely. That is a group of scientists which is saying it like this way, and they are also saying that the resistance, you don't require a pesticide for a genetically modified crop. Is that clear? That is true. You don't require any pesticide. It means the plant is producing toxin which is harmful for the insect. You are saying, means scientists are saying that toxin is harmful for the insect but not for us. What is the guarantee that won't be harmful for after the 50 years? To me, there is no guarantee. One group of the scientists is saying that toxin will keep on accumulating in our body and that will become harmful one day for our body. That's what is the argument, right? That's what is the argument, right? Clear, Affaf?
Yes.
Okay, so let's go through with it. The pharmaceutical and food industries now use genetically modified organism, also known as GMOs, to make a range of useful chemicals. However, as you have seen, prokaryotes do not process the biochemistry to make some of more complex human protein, but scientists have introduced useful human genes into eukaryotic cells, including the yeast, single cell fungal plant, and some mammals. The transgenic plant grow vaccines and may become the important weapon in the worldwide fight against the disease. That is beneficial for us. Scientists also hope that the modification of the plant will help us to overcome the challenges of the climate change and make possible to grow enough food of the right nutritional value to feed the overgrowing world population. That's what we discussed right now. In this section, you will look at the some of the more example of the ways which people are using genetically modified and organism and they consider the some of the risk and benefit of the new technology. Now GM, genetically modified crop. The plant, particularly cereal crops, are main staple diet, food normally eaten of the most of the human. The problem is that these crops are easily harmed by the disease, pest, poor growing conditions, and natural disasters, such as flood, drought. Traditional plant breeding has resulted many improvement, but it's a slow process. Means hybridization is a slow process and do not have that much benefit. Genetically modified can introduce a many useful characteristic much faster. Many of the first commercially produced GM crops were modified to make the life better. A producer, the seller food, for example, the growing longer shelf life, the uh, uh, the herbicide resistant, the big second wave of GM plant deliver improvement for the consumer, such as improving the nutritional content and the food and the making the crops resistant adverse condition. For example, including following. Do you know that is the BT cotton and BT brinjal? Have we discussed that? Affaf?
No.
Now, before going into that, I want to tell you what is the BT cotton. Actually, BT cotton was the first crop that developed in the world commercially. So let me show you what is the BT cotton. Then that will become more biotechnology application. Cotton. Just a minute. Give me 1 second. Mhm. Look, what used to cause, what used to happen, Faf? Previously, we have like a cotton ball worm. There was a cotton ball worm, right? So cotton ball worm. Let me explain you like this way. So previously, what used to happen? The farmer used to grow brinjal and cotton, right? So first take the example of cotton. Yes. So what used to happen, Affaf? Uh, like farmer used to grow cotton, right? Cotton, but there was a worm. What that worm to? There was a worm, and the name of the worm was cotton bollworm. So cotton bollworm, what it used to do? This was a worm caterpillar-like organism. What it used to do? It used to eat 50% of cotton ball. So farmer were very puzzled with this thing, and by this way, it reduces 50% productivity. But so this worm was very dangerous for the cotton, right? Cotton plant. It used to eat the cotton buds. Then scientists want to like what farmer used to do. Farmer used to give some insecticide, pesticide, and all that. Then scientists came to know there is a bacteria, right? The name of that bacteria was Bacillus thuringiensis. What was the name? Bacillus thuringiensis. Bacillus thuringiensis, it have a gene. The name of the gene was cry gene. So what was this? We given a short name BT, Bacillus thuringiensis. Got it? BT. What is BT used to do? BT used to kill the cotton worm. Kill cotton worm. How? Because it have a toxin and the name of that toxin was BT toxin. Did you got my point? Affaf?
Yes. Yes.
So who's the
B2
enemy of
toxin?
ball? Sorry. Yes.
BT toxin?
Yeah, Bt toxin. This Bacillus thuringiensis was a bacteria, right? This Bacillus thuringiensis bacteria, it used to go inside the gut of this bollworm and used to produce a toxin, Bt toxin. And this Bt toxin used to kill the bollworm. Got it?
Yes.
So, our enemy is cotton bollworm. And who's the enemy of cotton bollworm? Bt. So, who's my friend? Bt is my friend because he can kill the bollworm. Got it? So what
Yes.
scientists did, scientists take out a gene which used to produce the Bt toxin, and the name of the gene was cry gene. Got it?
Yes.
Now what scientists did, scientists taken the cotton plant cell, right, and inserted cry gene, right, where cotton plant. Now we called that new cotton plant Bt cotton. Now the cotton plant itself have Bt gene. Cotton plant itself used to produce Bt toxin. Now cotton bollworm could not eat the Bt cotton because it already have Bt toxin, right? And this cotton is called Bt cotton. Did you got that point? That's how we produce a Bt cotton. Then Bt tobacco and Bt brinjal. Got it? Affaf, what is the story behind the Bt cotton? Clear?
Yes.
Okay. Now come to your PDF. If you want, you can take the screenshot. Bt cotton, if you want. It have cry gene and it produces Bt toxin gene. And Bt toxin that eat it.
Done, sir.
Done? Okay. Uh, okay. Now the flood resistant rice. Now the flood resistant rice, complete immersion water destroys the rice crop. Right, during the flood time, the rice crop used to destroy. Right, because the plant used to get submerged, destroy the crops. But the global warming is causing more serious flooding. Scientists have developed a genetically modified rice that can resist being totally covered in the fluid water up to 3 weeks and still produces around 80% of the normal quantity of the rice. These went these went from lab field for 2 years and already preventing the people from the dying because of the lack of the food in those areas. Got it? So
Yes.
that was benefit. Now what we discussed, Bt cotton, pesticide resistance. Now Bt cotton, you have made the Bt cotton. Now you need not to put any medicine, any insecticide, pesticide against this, right? Now the plant is itself resistant. You made the plant resistant against cotton bollworm. Pesticide resistance globally, 20% or more all crops grown are lost to pest. If we could reduce this, there would be a lot more food everyone. Genetic modification of the crop plant can produce plant that makes their own pesticide within their leaves. This means farmers do not need to use the chemical pesticide which are expensive and can harm the environment. However, there are concerns that the pesticide genes will spread into wild plant and insect and fungi will become resistant to the plant chemical. That is also a threat. Changing nutritional value in the plant by genetically modified crop plant, scientists have the potential to change the balance of different chemical in the crops. If we can increase the vitamin, protein content of the food, like golden rice we discussed, or change in the balance of the fat, we can greatly increase the nutritional value of the food and the people who eat it. It may even be possible to grow the plant that contain the vaccine against the deadly diseases. That is the benefit of Genetically modified soybean, GM soybean. Soybean, known as soybean in USA, known as uh, soybeans in USA, and the biological name is glycine max, right? That are a major crop globally. They are also grown as a source of food rich protein for people and source of oil or commercial use in food industry. They are used in animal feed and about the biofuel which have been used to cosmetic industry. It is estimated that 90 to 95% of all soybeans grown in United States, a major producer, are now genetically modified. The scientists have used techniques including A tumefacients, agrobacterial tumefacients, and that guns to add a new genetic material in soybeans. That means genetically modified soybeans include that following: herbicide resistant soybean, means fatty acid balance, it maintain the fatty acid balance and all that, we incorporate that gene, right? Herbicide resistant soybean production is badly affected by the competition with weeds. So farmers spray a herbicide to destroy the weeds. However, soya plant are also killed by many broadleaf plant herbicide. You know the herbicide uh, they destroy the nutritional plant or crop plant. Scientists genetically modified soya plant that make the resistant to common weed killers. These GM soybeans are used widely across the areas of the world which grow soybean and production have genetically increased result. Fatty acid balance oil from soybeans widely used food production, but traditionally soya oil contain relatively a little oilic acid and stearic acid. Relatively large amount of the linoleic acid, that is very these are the actually uh, amino acids which are important for our body. Linoleic acid oxidize easily, so soya oil tends to oxidize no longer uh, uh, and good to eat. Means if it is not oxidized for longer time, it is very good to eat. Right? Uh, it won't produce a bad odor. Also, linoleic acid is polyunsaturated fatty acid, but oilic acid is monounsaturated fatty acid. And studies have suggested that oilic acid is better for the human health than linoleic acid. Scientists have produced genetically modified soybeans that are radically different balance of the fatty acid with a lot of oilic acid and less linoleic acid. This is now this has now two benefit. Uh, the oil lasts longer and may be healthier, right? So that is the benefit of making genetically modified soybean. Gene therapy, right? Gene therapy is used for the humans to cure many of the diseases. It is very exciting to think that the future we may be able to block faulty genes that causes some devastating diseases. People, however, research necessary to find the effective treatment is long and not always successful. For example, scientists hope cystic fibrosis might be cured by inserting healthy version of CFTR gene into the cells of the lungs. You know what is the cystic fibrosis?
I studied this in AS.
Destroy the alveoli. Alveoli
Yeah,
become looser, filled with the fluid, right?
yeah.
And causes respiratory disease. And it is caused by dust and all that, right? Most gene therapy is performed normal cells, somatic cells. So it is known as somatic cell gene therapy. Even if that person treatment is successful, they will still transfer their faulty alleles to their children. A potential solution of this problem is alter the germ cells so that the faulty gene are no longer is transferred. Right? Means alteration of the ova and the sperm. This include to be done early embryo immediately after in vitro fertilization. The new individual would be free of the disease, and children would be not risk of inheriting disease. Means inheritance of the diseases can be allowed, right? This may sound like a good idea. Some people argue that if technology is available, it should be used to benefit of many people as possible. However, many other are very concerned about the possible risk of the some people are offended by the idea itself. It is still not a certain what effect action could have on the early embryo, and the impact of the individual might not become clear until much later. It seems very positive to him. We are nowadays we have a successful technology that is called genetic counseling. What is the genetic counseling? If you'll take my gene and if you'll send it for the genetic counseling, they will make a profile, and on the basis of my gene, they can tell you that if I have any genetical disease, that would be transferred to my children or not. And if I have defective gene, they will tell you this gene is active, which is defective, and which will produce any disease. So what can be done before passing this gene to my children? They can alter gene this. That is called gene editing, right? So first it goes for genetic counseling, and that's what is mentioned over here. It seems very.
Sir, as
Yeah.
far as I know, it's not very effective. Sometimes the system makes mistakes.
That can be, but nowadays, right now, this is under research. So there is no something final final solution for the humans, because to do the research in the human, even in biotechnology or in any technology, it's a very it's a restricted, and there are a number of the restrictions on it. You cannot do experiment in human in easy way, because this may become for fatal for the humanity as well, right? So every government across the world are very concerned about these kind of experiments. So that is also a limitation for these researchers. Uh, it seems a very positive to aim or remove the risks of genetic disease, but it could be difficult to know. In future you will come to know that when you will become the old, you will see that the person which is sitting beside you may have the 50% artificial organ, artificial heart. There may be the person do not have heart. There may be a motor. Maybe a person do not have organ. Maybe that do not have certain like liver, do not have kidney and all that. Like means
Right.
there are fair enough chances the person may become 50% of the organs are artificial, means 50% human, 50% machine. That is quite possible, and these kind of the people are called cyborg. That research is going on. This year in the starting and the last year I was teaching about the cyborg in that is the UPSC classes. So that is type of the classes which goes on for the which make Indian top bureaucrats, right? So there is a competition. Do you know that UPSC? There is a competition which make UPSC in India. This is in fact this is the toughest competition in the world. UPSC that makes bureaucrat in our country in India. So there is a part of science, tech, and environment ecology. I teach most of the topics. So I was telling about the cyborg. We teach these kind of things. So cyborg is the future, future. And have you seen that there is a very famous movie in Hollywood? What was that movie? Uh, that movie has been made on the World War. I forgot, just slipped out from my mind. I forgot it. So that has been mentioned. Like cyborg, that was the first, in fact, cyborg. That movie was made on the cyborg. This this may sound a good idea, but okay. It seems a very positive aim to remove the risk of the genetic disease, but it could be difficult. It could be difficult uh, at what point to stop. Some people could prepare to pay a great idea of a great deal of the money for unethical treatment. For example, it raises the serious question about the manipulating the genes. It is simply remedy disease, but enhance the longevity, skin color, increase the adult height, intelligence, and all that. That is the risk actually. You know, one more thing. There is one more topic. Cyber was another topic, and we are also like what was that? Uh, we are incorporating the finer gene in the people in the human being, right? So that was also a topic where any manipulation of the bad technology can make a different race of the more intelligent people, more smart people, more strong people, and all that. So that may create a race of the human. So that is also a danger. Now you can think of every time if you are incorporating good genes and removing the bad genes, it means you have got a person, a boy or the girl, who's resistant for every kind of the disease, very intelligent, a perfect height, body balance, a perfect brain. And what else you want? He's like the girl or the boy, he or she is the next to the god. And who will be able to produce those kind of the children? Those who have lots of the money, they will be able to produce a fine kind of the generations. And those who do not have money, or they have the lack of the money, scarcity of the money, what they will do? There will be a different class. So there will be two class of the humans in this earth. So that is also a risk of bad technology. Risk and benefit. There have been a great deal of the public debate all around the world on GM technology. In the sum of the part of the world, Americans and Australia, people have a few problems with the use of the GM plant and food. In other areas, including UK and many other Europe countries, there is still uncertainty. This area of the science is very complex, and many people do not understand it fully. Uh, Much of their knowledge comes from the media, and most of the journalists, even the science correspondents, are not the scientists. It is not easy to obtain the evidence, nor it's easy to understand. Science is full of the uncertainties, but people like things to be clear and simple. As a result, many people, including the some of the politicians who make laws about the researchers, are going without all evidences, often with the incomplete understanding of the science. Uh, Interestingly, the use of the GM bacteria to produce human medicines such as insulin raises relatively a little debate. In case of the benefit of many people are very clear, and there appear to be very few risks associated with it. However, genetically modification of the food, plant, animals, and the humans divide opinion. Issues often are raised about the GM technology, including the following. In early days of the genetic modification, antibiotic resistance was often used to marker gene. People were concerned about the risk of these genes. Antibiotic resistance might get into the wild plant population and add problems. Antibiotic resistance. Scientists listed to these concerns and now widely use different marker genes, fluorescence or particular nutrient requirement instead of antibiotic resistance genes, right? So scientists have come out with the problem. Instead of as a marker gene, now the antibiotic genes are not used, only fluorescent genes are used. Concerned about the fertile seed. Scientists usually include the marker genes GM plant to make it easy identify them. These marker genes include the alleles that ensure the plant is infertile, so that the plants cannot reproduce. This removes the risk of them spreading into the environment. However, if it does mean that the farmer must buy the fresh supply of the transgenic seeds each year, planting the mm, planting. Uh, uh, Planting. This is more concerned the cause of the food crops than the plant which are being modified, which are modified and used as the medicine. But some issues raises. The big worry about this will be seriously disadvantage in the people of the poorer country. Did you got this point or not? Affaf, tell me.
No.
Now, look, what happens, what I told you, that suppose that I am a rich farmer, right? And we uses genetically modified crops, GM crops. We uses every time, right? Uh, I grow, like my family grow GM crops, right? And these GM crops, what they will do, so they will start the pollination, and the poor farmer which is using the original variety, they will pollute their variety as well. And gradually all the varieties, due to the pollination, because you can't stop the pollination, that pollination goes through the wind, and with the help of that, it will every year it will spread, and at the end of the day, what will happen after 10 years, what will happen, across the village there will be no wild variety, every variety will get polluted. So what scientists did when it happened, what scientists did, what they said, that this plant, this plant will be infertile. Means you will grow the seed, now the plant will be infertile, it will not produce any pollen grain. So what happened, this time I have grown a seed, right? I have got the crop. Next time I have to again purchase the genetically modified seeds. Every time this genetically modified seeds, they will, their cost will increase, and I won't be able, the day will come when it will become very costlier for me to afford that, right? And if any poor if any poor farmer want to do that, he won't be able to purchase those seeds, right? So that is the drawback of this one. Got it?
Yes.
To this point, explain this one. Worries over the risk of eating alien DNA or GM plant food. People forget that we eat many different type of the DNA every day and have enzyme specifically to digest, right? We may even eat the organism donated a new genes away. For many people in the risk benefit balance shift dramatically when GM contain a vitamin, medicines or important vitamin. In case of two, there are growing evidences that eating GM food carries no risk. Actually, the benefit is so the time may no longer be causing the concern, right? So some of the scientists says that there will be no longer concern about these GM crops. The concern which are present right now, they will remove it gradually. Environmental comes about the risk of the gene transfer of GM plant animals to the wild species, what I was discussing. As seen above, the people worried about the risk of antibiotic marker gene and other alien DNA spreading into the wild population causing the problem. As we collected more evidences that there are relatively little contamination of the wild plant by genetically modified crops, concern diminished. Right now, the scientists have developed that kind of the plant which have a little concern about the polluting the wild varieties with the, through the pollination. A strong objection to use the animals and other organisms for the scientist research. Some people feel that genetically modification threatens a right to modification of the organism. Some people believe that there is no benefit of people or more important than the risk of animals of being used to scientific researches. Right. For scientific researches, we are using the some animals. We are doing the experiment that may become harmful for in future and sometime. Like you can say, if the coronavirus was an experiment and it was a research, how fatal it was, you can realize. That was also a biotechnology part, right? Do you believe that was a actual virus or it has been made? What do you think?
I think it was
Coronavirus.
me.
It was made, right? And it was made by China. That is true. We strongly agree, and everybody knows that. You must have listened that we know that this person, what to name his was, what was his name? That U.S. president uh, before the Biden, Joe Biden.
Oh, Trump.
Yeah, Trump. Trump says, don't say it coronavirus, say it Chinese virus, right? And he was very straightforward kind of the person. That is sometime it is said that ideally that is not expected from the US president, but what he said strongly, that was true. Everybody knows that was the virus which was made by China. So that kind of the experiment of the biotechnology can become the fatal for the world. That is also a concern. Real concern that gene technology will uh, will be property of few companies in developed countries. That is also true. Some people fear that the new advance may be largely biased toward the need of the richer countries. However, work on the plant-based vaccines and food and uh, flood and drought-resistant rice are being made available at very low cost in the place or in the places it is needed, which is helping to counter and fear ensure the benefit of GM technology. So some people have questioned that it will may become beneficial for richer country. They may hold the technology, but nowadays what is happening that these richer country are doing the experiment like flood resistant, drought resistant plant, and they are providing it to poor nations as well, right? So this myth is getting clear. Today, the use of transgenic plant medicines such as vaccines are relatively new and unknown. So the concern about the risk in the general population are at low level. Perhaps if the media coverage become negative, worries will grow. That is true. However, it seems likely that same way using the genetically modified microorganism to produce human medicines, people will accept the benefit of this technology, uh, seeing those benefits as being greater than any possible risk. As scientists' evidences build, the scientists increasingly convinced the safety and the value of GM crops. However, with the other GM food, some people still have concern about the possible risk for the humans uh, that use them and for the animals that are used to produce them. Despite of this, for the great majority of the world, food security is often issue and infectious disease, malnutrition still kill the huge number of everywhere. GM crops and medicine may will offer the difference between the survival and the death. Right? They'll help the people to survive and they will reduce the chances of death. So chapter shows there's some of the areas concerned about the gene technology which are being debated. Despite of these concerns, many people, including the most of the scientists, feel that the enormous benefit resulting the GM technology development of food production and medicine development and potential therapies are much greater than the any risk of the ethical obstacle which are arising. When people express concern about the risk of using GM organism in medicine food, it's wise to remember that a risk can come from the wide variety of the sources, including the starvation, eating the poor quality of the food, infectious disease. In the future, hope is that the GM organism will help to reduce the sum of the risk and making them part of the solution, not the problem. So, in the nutshell, what you have to do, you have to write it down the on the like point basis on the bullet format. You have to write it down the benefit and the risk of that one, right?
Benefit and risk of what?
Benefit and risk of biotechnology. You have to
Okay.
jot down the point, whatever
That's
we
all
had.
from this.
Yeah, that's all from the chapter.
No, it was easy. Not too
Sorry.
much. It was easy and
Yeah,
interesting.
interesting. I told you that this is the most interesting part of the genetics and biotechnology is the most interesting part of the biology, at least for me. I love biotechnology. That's it. Your syllabus completed.
Yeah, except for the practical part, but because like I told you, I have 3 exams. June 1, which is Unit 4, Topic 5 and 6. June 5th is Unit 5, Topic 7 and 8. And then June 16 is practical. So between 5 and 16, we can finish practical easily.
Okay, so you want to finish the practical between the June.
I can do it before as well, but it's not